Welcome to the technical demonstration of the LifeWatch-ERIC Tesseract Virtual Research Environment IJI NIS workflows. We are going to start exploring the system by logging in. We type our LifeWatch EU user and the password. Once logged in, we get into the dashboard. In the dashboard, we can see Tesseract Beta. When clicking, it will open the IJI NIS workflows in a new tab. Once the page is loaded, we see the main layout. On the top right, we can find the user menu with the user account and the link to log out of the system. The first content that is displayed to the user is the personal space. This is an overview of all the data that the user has access to. And on the left, we have a list of all the workflows that this user has access to. This user has previously requested access to these workflows. Even though different workflows consist of different set of tools, all of them share three buttons in common: the Dashboard button, the Run a New Workflow button, and the Workflow Information button. Besides getting an overview of all the files that has been shared with the user can also edit, upload or delete files and folders that are in their personal space. The dashboard button of all the workflows is the place where the user can find the overview state of all the workflows that has been run previously or that are running currently. In the Run New Workflow button, you can execute a new experiment. Finally, in the Workflow Information button, you can see the workflow diagram and a scientific explanation about the workflow. You can also access the information page of each of the workflows. Let's start exploring the Ailanthus altissima mapping workflow. Combining modeling and remote sensing techniques to monitor and control the spread of invasive species. The case of Ailanthus altissima. Ailanthus altissima is one of the worst invasive plant species in Europe. It reproduces both by seeds and asexually by vegetative sprouts. The winged seeds can be dispersed by wind, water, and machinery, while its robust root system can generate numerous suckers and progeny plants. Ailanthus altissima typically occurs in very dense clumps, resulting from even-aged seeding establishment or from clonal expansion through root sprouting, and occasionally it grows as widely spaced or single stems. It grows on a broad range of anthropogenic to natural sites, from stony and sterile soils to rich alluvial bottoms. Due to its vigor, rapid growth, tolerance, adaptability, and lack of natural enemies, it spreads spontaneously, outcompeting other plant species and reducing their growth. In the last decades, the species is quickly spreading and multiplying in the Alta Murgia National Park, in the south of Italy, mostly characterized by dry grassland and pseudo-steppe, wide open spaces with low vegetation, whose tendency is to be easily invaded. Ailanthus altissima causes serious direct and indirect damages to ecosystems, replacing and altering communities that have a great conservation value, producing severe ecological, environmental and economic effects and causing natural habitat loss and degradation. This threat is likely to increase in the future, unless robust action is taken at all levels to control the advancement and spread. In a recent working document of the European Commission, it has been evidenced that the cost of controlling and eliminating invasive species in Europe amounts to 12 billion euro per year. Two relevant questions then arises: whether it is possible or not to fully eradicate or at least to reduce the impact of an invasive species, and how to do that at a minimum cost, in terms of both environmental damage and economic resources. The LIFE program, the European Commission Financial Instrument for Environmental and Nature Conservation, funded the LIFE Alta Murgia project, which has, as a main objective, the eradication of the invasive exotic tree species from the Alta Murgia National Park, and which provided both the expert knowledge and the infill data for the case study we have started. Actually, there is a single map of Ailanthus altissima presence, provided by the LIFE Alta Murgia project, and it is dated at 2012, before the starting of the ongoing eradication program. Due to the lack of data, predicting the extent of invasion and its impacts are extremely difficult, as well as assessing the efficacy of any control measures. If static models based on statistical fitting cannot predict any spatial temporal dynamics, in example, where and when the ailanthus trees may repopulate a zone, mechanistic models which incorporate growth and spread of a plant would require precise parametrization that is extremely difficult with the few available data. Due to these limitations, we rely on a relatively simple mechanistic model, a diffusion model, which is validated against the current plant spatial distribution deduced by satellite images. The effect of an eradication program is taken into account with a reaction term, which simulates the plant eradication during a control program. The value of our predictive mechanistic model would be to provide an automatic tool for an a priori estimation of the effectiveness of a planned control action under temporal and budget constraints. In our study, the interest is focused on finding the best budget allocation both in space and in time for the park area maintenance. This aiding in determining whether a control policy needs to be improved. Within the activities of the Ecopotential H2020 project, we developed an automatic tool that can get the plant presence information from the satellite and use that information for predicting the best action of the park manager. Indeed, many satellite data are not being used to their full potential, and there are optimization methods that are powerful but not being used on ground. We are linking the two methods and providing an avenue to make them accessible and useful to managers. We are currently estimating the uncertainty of the model and working at the validation of the overall approach by testing it on different areas, by using aerial images coming from a regional project. The final aim is to provide a robust tool to be easily transferred to other geographical areas and potentially to different species. The approach might incorporate the effect of changes, climate, land cover. This is still an open research issue, which will largely benefit from the exploitation of the LifeWatch-ERIC infrastructure. To run an Ailanthus altissima workflow, we go to the left menu and under Ailanthus altissima mapping, we click on Run New Workflow. The first thing presented to the user is a static diagram of the workflow. and an overview of the required files for this workflow to run. The first piece of information the user is requested to provide is a conceptual name for the new workflow. In this way, the user can identify and remember each workflow more easily. For instance, Monday morning experiment. In the next step, the user is requested to introduce all the seasonal data. In this case, we are going to use the dummy testing data. We select the multi-season images, the multi-season testing data, and the multi-season training data. In the next step, the user is requested to add the two-season data. This is the images, test data, and training data. Once all the necessary information has been provided, the user can create a new workflow pressing on the Create a New Workflow button. Once the system created a workflow, the user can run a new Ailanthus Altissima workflow or go to the dashboard. On the dashboard, we can see that there is an experiment running, the one we just executed. To get a better insight about the workflow execution, we can go to the detailed information of this workflow. In the workflow inside, we can find the general information for this workflow, which type of workflow it is, which name was introduced by the user, what is the current state, when was created and updated, and what's the progress of execution. In the working status diagram, the user can get an overview of all the steps that are running, finished, or waiting to start. In the workflow output files, the user can find all the files that the system generated for all the steps. In each one of the steps, the system generates a log file. At any moment in time, the user can refresh the status of this page. Once the workflow is successfully finished, the user can have access to the output information. For instance, the last step of this workflow is the extractor component. In the workflow output files, we can see the result for the component extractor. And we can download the output file and also the log. Let's continue exploring the system, taking a look to the next workflow. The ARMS workflow. The ARMS European project is a genomics observatory and a collaboration of 20 European institutes. Our network of autonomous reef monitoring structures, ARMS, consists of 25 observatories who deploy individual settlement units in the vicinity of marine stations and long-term ecological research sites in European coastal waters and Antarctica. After a few months, they are brought up, and visual, photographic, and genetic assessments are made of the life forms that settled on the dam. This data will be used to assess the status of and changes in the hard-bottom communities of these near-coast environments. This project began in 2019 and will run for as long as funding allows. The scientific goals are 1. To identify newly arrived non-indigenous species at each ARMS site. 2. To track the migration of already known NIS in European continental waters. 3. To monitor hard-bottom community composition over longer periods. And 4. To identify the essential biodiversity variables for hard-bottom fauna, including non-indigenous species. In order to run a new ARMS workflow, the user has to click into the Run New Workflow button on the left panel under the ARMS section. The first piece of information displayed to the user is again a static diagram of the workflow. This gives an understanding of the required files to run this workflow and all the steps that will take place. As in the previous example, the user introduces a workflow description to identify the experiment. The next step for the user is to select the input genes for PEMA. For doing so, we developed a new tool. This tool displays the genetic samples on a map and the information about the samples on a grid. This tool can be made full screen for better filtering. And the map can be displayed or hidden. In order to help the user relating the grid information with the map, we developed a Show on Map tool. When clicking on it, the map centers and zooms on the selected sample. When the user knows which gene or genes wants to include as PEMA parameters, she or he can select them from the grid by just clicking on them. The selected genes are displayed at the bottom. This time, instead of selecting dummy testing data, we are going to try to upload our own data. The user can create a folder and then upload the data. Once the file is uploaded, the user can select the uploaded file as an input data. The next step is to introduce the parameters for PEMA. This can be done manually introducing them one by one in the form, or uploading a parameters file. The next step for the user is to select the RIMS parameters. This is a latitude, a longitude, a latitude radius, and a longitude radius. To make it easy for the user, we developed a map where the user can zoom into a specific area and drop a marker. The marker will automatically get the latitude and the longitude position. The values can also be filled in on the right form. The last step is to click on Create New Workflow. Again, the user can see the inside of this running workflow by clicking on Detailed Information. Once again, when the workflow is successfully finished, the user can explore all the output data by component. The user can also then download the final result. The next workflow that we are going to explore is Biotop. Vulnerability workflow. Risk assessment of non-indigenous species introduction and establishment. Habitat vulnerability to non-indigenous species and estimation of the impact on biotopes. Assessing ecosystem and habitat type vulnerability to invasion, colonization of non-indigenous species. Is key to focus conservation actions protecting them from the negative impact related to non-indigenous species invasiveness. This validation case aims at mapping ecosystem and habitat type vulnerability at continental scale, inferring the relevance of key risk factors, in example vector of invasion, and intrinsic resilience components, in example native biodiversity, food web structure, and design scenarios of change in the context of expected climate changes for ecosystems and habitat type found highly vulnerable to non-indigenous species. This workflow aims at highlighting, at the scale of Europe, where the incidence of invasive alien species is the strongest and what areas or habitats, when possible, are the most vulnerable to the negative impact of invasive alien species. At present, the scale considered is the one of European countries and respective marine regions. To run a new biotop workflow, the user can press on the button Run New Workflow under the biotop section. Once again, the first displayed information is a static diagram of the workflow, followed by the workflow description. And the button to create a new workflow. Once the workflow is created, it will be visible on the dashboard. And the process of executing the workflow can be followed in detailed information. For the biotop workflow, an extra tool has been developed, the Result Explorer. We can access it under the Biotop section, clicking on Result Explorer. This tool helps to analyze the output incidence data from the workflow. Additionally, the user can access to all the metadata information from the file, clicking on Expand All HCIF Metadata. The next workflow to demonstrate is the crustaceans functional biogeography. Functional biogeography of invaders, the case of two widely distributed omnivorous crustaceans. Biological invasions are to date acknowledged as significant environmental and economical threat. Yet the identification of key ecological traits determining a species' invasiveness has remained elusive. One unappreciated source of variation concerns dietary flexibility and the ability to shift trophic position within invaded food webs. Trophic plasticity may greatly influence invasion success, as it negates resource availability as a functional constraint for introduced individuals. In addition, the impact of an invader might increase with its dietary plasticity, since a greater proportion of the resident assemblage of a species and multiple trophic levels are affected. The validation case will focus on two invasive crustaceans widely distributed in marine and freshwater European waters. In example, the Atlantic blue crab, Calinectes sapidus, and the Louisiana crayfish, Procambarus clarkii. The key questions that will be addressed are: A. Does the trophic position of C. sapidus vary between invaded Mediterranean and native West Atlantic habitats, testifying the occurrence of significant post-invasion shift in dietary habits? B. Does the trophic position of P. clarkii vary in invaders' habitats, and which are the main ecological factors involved? C. Do bioclimatic drivers influence broad-scale spatial patterns of variation in the trophic position of C. sapidus and P. clarkii? To run a new crustaceans functional biogeography workflow, we go to the left menu and find the right section. Under Crustaceans Functional Biogeography, we press Run New Workflow. Again, the first piece of information displayed to the user is a static diagram of the workflow. As in the previous example, the user introduces a workflow description to identify the experiment. Next, the user is asked to introduce the biological data on CN stable isotopes. Followed by the trophic position data. The last step is to press Create New Workflow. The process of the execution of the workflow can be displayed in detailed information. Finally, the result of the last step of the workflow can be downloaded. An additional tool has been developed for this workflow. It allows to the user to manually validate the samples that come from the algorithm of this workflow. To see how the tool looks like, the user can go to the left menu and click on Geographical Validation. The Geographical Validation tool is designed to efficiently visualize all the samples that have been input by the user for this workflow, and to filter in or out all the samples that the user considers valid or invalid. To do so, the user can visualize all the samples displayed on a map. At the bottom, there is a table containing all the information of the sample, such as the data that was collected and the description of it. To help the user with the visualization of the samples, this tool can be made full screen. Also, the different sections can be expanded and collapsed. The map has a built-in functionality to allow the user to zoom in a specific area by selecting it using the mouse. The user can mark a specific sample as valid or invalid using different tools. The first method is done by selecting one or multiple samples to validate, and then selecting the toolbar on the right to specify whether or not the selected samples are valid. The second option involves exploring the table of samples. A specific sample can be located on the map by clicking on the Select on Map button. The user can mark whether a sample is valid by clicking on the corresponding checkbox. Once all the manual filtering has been completed and the user has made sure that all the data has been reviewed, the Continue the Workflow Execution button at the top of the page can be clicked on. The last workflow we want to demonstrate is the metabarcoding workflow. Validation case: detection of non-indigenous species with metabarcoding, for example, freshwater fish. Objective: the main aim of this tool is to detect likely non-indigenous species with metabarcoding. Detailed questions that will be answered with this tool and for each metabarcoding sample are: 1. What was the taxonomic resolution achieved with the marker, species, genus, family, and pipeline used? And how many OTUs were non-attributed to any taxa? 2. Which taxa were listed? 3. Which species are listed as non-native invasive in the checklist for the country published by GBIF? 4. Which species are not listed as non-indigenous species? In example, native in the region country or new detected taxa? 5. What is the number or reads of each taxa in the new NIS list? 6. How much does the species not listed as non-indigenous species overlap with its known distribution area? To run a new metabarcoding workflow, the user can press on the Run New Workflow on the left menu, in the metabarcoding section. The first piece of information is a static diagram of the workflow, each one of the steps, and the input data. Next, the user can introduce a workflow description. In the third step, the system asks for the occurrences data. When the occurrences data file is selected, the last step is to create a new workflow, pressing the Create New Workflow button. Once the workflow is created successfully, the user can see it executing in the dashboard and can check the execution process going to Workflow Insight. Once the workflow has been finished, the user can inspect all the output data by all the components and download the output data of each one of them. It is also possible to see a detailed output of the execution of each one of the components by exploring the task list details. During the development process of all these tools, reusability has been taken into account. This allows us to reuse the components that we already have been building and create tools of the future. For instance, this is an improved version of the Workflow Studio. Here, scientists will be able to select different components from various workflows and create their own workflows